Okay, a very warm welcome to everyone, and thanks for joining us for the webinar today. As a brief introduction to myself, I'm Paul Thor. I head up structural sciences here at SPT LabTech, and it gives me great pleasure to introduce Professor Mike Probert, who's head of structural chemistry and the crystallography facility at Newcastle University in the UK. As a bit of brief background on Mike, he obtained his MChem from Oxford in 2001 and completed his PhD at Durham in 2005. Mike's been at Newcastle working with Michael Hall there since 2013. As a brief overview of the webinar, Mike will give an introduction to Enact, this encapsulated nanodrop crystallization technique, which we've been developing over the past years. He'll discuss what its key benefits are, detail some success stories, highlighting the structural diversity of what it can be applied to, and also let you know how you yourself can access this new method. A few housekeeping rules before we get started. There will be a Q&A at the end, so if you do have burning questions for Mike or myself, please post in the questions panel and we'll deal with those at the end of the presentation. So it's over to Mike for what hopefully will be a fascinating talk. Thank you, Mike.
Okay, many thanks for SPT LabTech for organising this webinar, and thanks for joining. I guess it's good morning, good afternoon, good evening, depending on where you are in the world. So thanks very much for coming along. I'm Mike Probert, and I'm going to walk you through some of the crystallization approaches that we've been developing over the last few years. If you have questions, I'll happily take these at the end. If you have questions that come up after the event, that's fine. My contact details are all in the top left of each of the slides, so there's plenty of time to note that down if you've got questions. Okay, so we're going to look at high throughput nanoscale crystallizations, particularly around small organic molecules and pharmaceuticals, over the next half an hour, 40 minutes or so. So I guess we'll just get straight into it and see how it goes. So really, the first place to start is why should we worry about crystallizing molecules at all? And this could be for a range of reasons. We might be thinking about process development, purification. That's probably the reason most of us came across crystallization in the first place, when we do a recrystallization in undergraduate laboratories to really increase the purity of our compounds that we've made. But it's not the only thing. We might be trying to tune particle size and shape. We might actually just be interested in trying to work out what we've got. So most analysis of chemical compounds is done by spectroscopic methods. But if we can grow single crystals of our material and use single crystal diffraction with one of many different radiation types, then we get access to the full molecular structure, the full crystal structure, and the full stereochemistry of the system as well. So from an analysis point of view, this gives you almost a catch-all of the information that you need. But also it might help in terms of formulation, the stability, the bioavailability of a compound, all of which can be tuned just by changing the crystallization process and the ingredients that go into that. So we've got this kind of pictorial representation of this, starting off with some compound in solution, going through some magic system of crystallization to give some crystalline sample that we can then either use for analysis or for taking through in process development, or from actually a formulation point of view into our compounds that we want to take forward to market. And the crystal form that we get, so once we've done this crystal growth, we can also analyze it against different conditions and set up the chance to analyze different polymorphs, where we've got the same compound giving different crystalline forms, which might have different physical properties, and the same being true for salts, co-crystals, hydrates, solvates. The raft of solid state materials that we can approach at this point is large, if we can analyze the crystallization space sufficiently. So well, how have we done this in the past? What's our base point? Where are we starting from? Most of us will have learned techniques kind of around these ones in the center, particularly around evaporation or cooling. So starting off from a hot solution and cooling it down and seeing our material drop out of solution, or evaporation where we have a slow concentration effect and we reach a suitably saturated position where the material can no longer exist in the solution phase and forms our solid. And we can look at either cartoons or mock-ups of this system on the left, where the amount of material we're using is relatively large. The scale of the experiments is quite big, really dependent on the skill of the researcher as well. But also, these are single shot points, so if you want to map out a large amount of crystallization space, these manual techniques become prohibitively slow. They are incredibly flexible, but the amount of time commitment from the researcher is huge. So this is problematic if we're using this to explore crystallization space, particularly when the amount of material per crystallization experiment might be quite large. So we have some issues there that we need to be aware of, particularly if we're in a sample limited condition. There are some automated techniques which take these manual approaches and just speed them up a little bit. They are less flexible, require actually larger amounts of material, and generally give back microcrystalline and powder samples rather than the single crystals that we would like for full analysis. So really then, that kind of sets us up nicely, although there's been a kind of game-changing advent of technology in the recent years. So do we need to grow crystals at all? The cryo-EM stroke micro-ED community have blossomed in this realm, where instead of using X-rays as the incident radiation, we're using an electron beam. And this allows the size of the sample being analysed to be much smaller. And that's because the interaction of our electron beam is much stronger with our sample than the interaction of our X-ray beam. It's not without issues. It's much more expensive at the moment. The sample preparation is definitely more complex, and this is even more true for systems that have trapped solvent molecules within the system. Any loosely bound component has to be treated very carefully when exposed to the vacuum chambers that these approaches run under. Determining the absolute stereochemistry is a challenge. It's a challenge that's mostly solved now using analysis of dynamical scattering, but it is definitely more of a challenge than it is from single crystal X-ray data. And of course you still need single crystals. They are just much smaller than for X-ray diffraction analysis. I've put some scales on here for the size of crystals required for various places. For cryo-EM and micro-ED, you can scale this down significantly. But what about if we don't need to grow crystals at all? If we just want to analyze our compound, we could use the crystal sponge technique, sort of pioneered by Fujita, where you soak your analyte into a host crystal, and you use the cage-like structure to trap your compound in hopefully ordered orientations within the structure, and then when you analyze the host structure, you gain analysis of your compound as well. This has been used very successfully across a range of compounds where molecular information is gathered. The problem with this technique is that it doesn't give you data on the intermolecular interactions, the full crystal structure of your compound, because the compound is there as a host within, as a guest within the host. Now, that is slightly problematic, but technique is incredibly advantageous for samples where you're not able to access the solid phase under ambient conditions. So if you're a gas or a liquid under ambient conditions, then gaining a crystal is significantly more of a challenge. So I think both crystal sponge and micro-ED are techniques which we consider as parallel tools for problematic crystallization, and should definitely not be forgotten. Luckily, they're not in competition at all. These techniques are entirely complementary. Okay, so let's move on. Given that we know we want to ideally have crystallization, why are we stuck with these approaches that we've known about for decades and not really improved? Why can't we do better? The protein or macromolecular community have faced these challenges since that field's inception. The amount of material that the macromolecular community has to work with is very small. And the crystallization space is different but equally vast. It varies by pH and buffer type rather than the solvent and anti-solvent combinations or the coformers, but it is still a wide-ranging challenge. But they seem to have done quite well, so is there any way we could take some inspiration from that? Well, if we look at the general setup of what they do, the crystallizations that they set up are just miniaturized versions of the manual techniques that we know about and have known about for many, many years. So the question is, can we use the way that they parallelize their crystallizations for our small molecules that are soluble in organic solvents to enable us to access crystals? Because if we can, then we can start using some robotics, some much higher throughput. The same kind of multi-well plate format that is used will allow us to kind of process those things much quicker. We will reduce the amount of solvent we need for any given crystallization, and reduce the amount of material that we need by orders of magnitude. And these robots, one particular example would be the SPT LabTech Mosquito. So if we go forward with that and we just think that generally the process seems sound, how will it translate across? Knowing that if we can, our 1 milligram of sample that would have been one crystallization experiment before could easily lead to 1,000 experiments, each of which could give us a crystal, each of which could give us a single crystal structure. One worry we started off with, which turned out to be a little bit unnecessary, was the size of the samples being formed. If you actually do a back of the envelope calculation, we're still far in excess of the amount of material in each crystallization condition for analysis, so that's okay, we don't have an issue. If we did, we could always turn to micro-ED as a solution if we can form our crystals. A general incompatibility of the equipment with organic solvents, that is an issue, and across the community when we started on this project was seen as a showstopper. We'll talk about that in a second. And the third thing that we need to consider is the rate of organic solvent loss on a nanoliter scale. The protein world have much success in this because their aqueous media is generally relatively stable, generally not the case for organic solvents. So let's take these two stepwise. A small amount we'll talk about the equipment incompatibility. This is mostly founded around dispensing robots or liquid handlers having a reusable nature and rubber seals within the system to allow the dispensing of the sample. This isn't the case with the Mosquito robot from SPT LabTech. It has disposable dispensing tips that come into contact with any component of the crystallization for very small amounts of time. Even so, there was a little bit of worry over the materials and our organic solvents. Plastics generally don't survive well with aggressive organic solvents. So in collaboration with Paul Thor, we set up some tests. We took some of the dispensing tips and left them submerged in our solvents for a period of time. Turned out to be quite a lengthy period of time, and then had a control to compare against. And essentially we saw no degradation of the plastic over several days. There was no discoloration, there was still structural integrity, and we knew that the contact time of our solvents with the plastics was very, very small in any dispensing situation. It's of the order of seconds rather than the order of days. And because those dispensing tips are disposed of, there's no contamination from one system to the next, and there's no worry about degradation of the performance of the liquid handling robots over time, because they're or at least caused by the solvents. So our big issue then is this rapid evaporation of organic solvent on the nanoliter scale. So that's something we really do need to get a handle on. So can we think of a mechanism to slow the solvent loss? And this was our challenge. Obviously the fact that I'm talking to you shows that we managed to succeed. But how did we do that? Well, we had this idea that we ended up calling Enact, so encapsulated nanodroplet crystallization, where we would take a dispensing robot, dispense an oil droplet, an inert oil, and then inject into that droplet our analyte inside a solvent. So this cartoon across the top is really showing what we hoped would happen. So we get our initial injection, our concentration with either concentration by diffusion or evaporation with solvent leaching along the border between the oil and our glass slide, leading to a nucleation and then crystal growth that we could then extract and run single crystal X-ray diffraction analysis on. And this allowed us to kind of scope out that we could use varying volumes of our oils, we could use varying volumes of our analyte as well, although we've pretty much homed in on 50 nanoliters of a sample. And that's approximately 2 to 3 micrograms of our analyte in question. And then we nominally seal the whole system with an extra glass plate on the top, which allows us to examine all of the conditions by optical microscopy, and it allows any extraction of our sample to be done and analysed by single crystal X-ray diffraction. So that was our cartoon mock-up. So how well did it perform? Well, surprisingly well. I haven't shown an oil droplet on its own here, but if you imagine a blank droplet here, then we have our injection, our concentration, our nucleation and start of crystal growth, and our crystal growth to size, which is large enough for us to operate on. And you can even see the change in colours showing the difference in concentration of our solution trapped within these inert oils. And if I show this in a time-lapse video, this is a slightly malformed droplet where the analyte has drifted across to the border of the oil, but you can still see a boundary of oil around it, and we can see crystal growth within this droplet and changing local concentration gradients within these droplets for this moderately simple organic system. And one of these crystals on the top edge was more than large enough to run in-house. So if we move on, we look at our first test set of compounds. This is one of them, you'll see it on the next slide. And then see how well this has worked. So we did cheat, I will openly say. We started off with compounds we knew were crystalline because we wanted to stack the odds in our favour a little bit. And we went through with some things which are pharmaceutically related. We have aspirin and naproxen. We've got caffeine, a nice additive, and then some organic systems. In the central columns of these you can see real data from the analysis of the structures. So these are FOBS maps. So this is the electron density that is seen for our structures, getting very, very good localization of density. So the crystal quality is very high, and then a final refined structure on the side with all the hydrogens taken out. And you can see even resolution of the stereochemistry via the FLAC parameter is very good. So this generally works. So does it work on this subset of compounds? Absolutely, although this subset of compounds was stacked in our favor. But what we wanted to do with this set of compounds was look at whether or not our approach was making things better. So we applied a bit of statistical regression to these systems. So we took our 5 test cases, we tried them with different solvents and then different oil encapsulations. So this top row here has no oil in encapsulation at all, and then we have 2 different types of oil on the bottom 2 larger rows. And then within each one of these solvent systems we have our 5 different compounds, our analytes, and then essentially the way to interpret this image is the more green, the more successful the crystallization was. So even with no oil encapsulation, some systems crystallized reasonably well, suitable for analysis. But as we progress through to our encapsulated systems where we've slowed down the evaporative loss, we definitely have an increase in crystallization success. And interestingly, at this point, there is a different success rate for combinations of oils with different solvents. So the combination of oil and solvent is very important. So the oil is not quite as innocent in this as we might think. We developed a categorization system for relatively rapid analysis, and then using this categorization system we could prove that the encapsulation with oil actually improved the crystallization outcome. If these peaks were centered around 0, it would have had no positive impact. If we have a value in the positive direction, this shows us we have an increased effect on success, and the counter argument would be true if it was negative. So that was with our first test set. We expanded this range, starting off with some, again, simple molecules, but going through to some things which were moderately complex, so vitamin B12, the largest of the vitamins, and gaining good data even for systems that are really quite complex and include metal chelation within the system. We can go to things which are flexible, so cholesterol, having many rotatable carbon bonds within the structure, showing that this is actually a system which can work for a broad range of chemistry and chemical entities. So we can show that the technique works from a straightforward crystallization point of view for our small organics and pharmaceuticals. So if we were to take that and run with it, can we go beyond just crystallization analysis and looking for our result and interpreting our structure? Can we start exploring polymorphism? Can we look for changes in structure of the crystal whilst keeping this compound the same? And if we set up using the kind of king of crystallization, this red, orange, yellow Roy structure that's been analyzed over many years, and we set up a crystallization plate, we can see that we get crystallization of many different colors within the spectrum that we're expecting, and very many different morphologies. And all of these can be found within different conditions. Some grow concomitantly within the same well, so we get different polymorphs within the same conditions. That's to be expected. The energy differences between these polymorphs is quite small. But if we do this analysis and we go through and we look at all of the structures that we get, we actually access all of the known forms at the time of publication, anyway, all of the known forms from the solution phase, this yellow, orange needle, orange-red plate, the red crystal, and we found another polymorph of this R18 sample. We'll circle back to this R18 in just a couple of slides. But this shows immediately that we can do polymorph exploration relatively quickly with relatively small amounts of material. On further analysis of the crystallization of Roy, though, we were also able to find this Y04 system, the Y19, which have previously only been crystallized via different approaches. So this has only been accessible via the melt or seeded with a related compound. Whereas Enact seems to be giving us access to these different states without this need of approaching the melts, and also gave access to yet another new polymorph. So this kind of returns Roy to its absolute leader of the polymorphism realm. So this is currently in preparation for a publication. So if we just circle back to that R18 form, this one here, this turns out to be highly metastable, and this is where I just want to highlight that using those techniques we described earlier can be particularly beneficial. So that R18 form can undergo a transition either by pressure or temperature stimulus and goes to a predominantly amorphous phase. And this is an SEM image of that amorphous phase, and you can see, although the majority of this is amorphous, there are rod-like crystals growing out various places. And micro-ED allows us to do analysis of those small rod-like crystals, which you would never be able to analyze by single crystal X-ray diffraction, and does give a solution, although does suffer from some of the problems that micro-ED generally has at the moment, which is higher than we like to work with R factors, so ones that are not so common for X-ray diffraction, but is clearly showing us the material and actually giving us a strong indication of what that system transforms into. So we've seen then that we can generally use this technique for crystallization of compounds, and we've seen that we can use it for analysis of polymorphism. What about if we wanted to do systematic studies, though? Because we now have a highly parallel crystallization approach, we can start analyzing compounds to look for trends across the systems. So again, for an easy starter, we started with a class of compounds that we were likely to get crystallization from, but we looked at the small organic acids, and crystallization of all of these compounds is easily achieved with large high quality crystals grown, all of which are suitable for analysis and all of which give good results. So this changes the way that we might think about crystallization to not just a tool for analysis of a compound that we are particularly interested in, but into a mechanism that we can analyze a class of compounds to try and work out if there are crystallization trends that we might be able to spot at this point. So this work is currently in preparation for another publication. We can also do the same thing with co-crystals and looking at co-crystallization type experiments. But what about samples that are classified as, in quotes, uncrystallizable, and things that people have struggled with in the past? So we were posed this question as a commercial case study, really. So dithionon, so this is a broad spectrum fungicide. It's been in use for many decades, actually. So it's been on the market without full structural analysis having been done from a single crystal data set, and the structure was predicted from powder work done several years ago. And we were proposed this as well. If Enact works in the way that we think, can we get access to single crystals of this nominally uncrystallizable compound? And there's some experimental conditions set up on the side, but in the interest of time, we won't go through those. We'll just skip through to the headline act, really. And that is that, yes, we can. So we get these crystallizations resulting. Good success rate in that within a 2-week period we had access to different crystals that we could run and actually gain full structure resolution from this. So from a commercial point of view, compounds that are difficult to work with, have proved difficult to crystallize before, this technology seems to work very well for. If we think about academic research, this is a project we did in collaboration with Anne-Marie Donoghue in Durham. She had this series of compounds, and the 3 highlighted in red boxes were the only 3 that they could not get to crystallize and had limited amounts of material to work with. So in a very short period of time, again within the 2-week period of being working with the samples, we had access to all 3 of the missing structures, which completed the set and allowed her to progress with publication. So we can definitely use this approach on systems which have classically been difficult to crystallize. Another one of those types of compounds that we might think about is in the natural product world, where larger amounts of flexibility and sample quantity limitations are definitely present. So in collaboration with Professor Nick Oberlees, we've approached a large number of these different natural products now and have had good success. The success rate here is slightly lower than it is with the crystallization of our classical small molecule systems. But if we look at the total number of compounds we've analyzed, we've looked at around about 60 different natural products, each of which we generally have less than 10 milligrams of the material. That's essentially the world's supply. And about half of them, almost exactly actually, have resulted in full X-ray structure determination and mostly resolved stereochemistry. And this is a much higher hit rate than normally would be expected for the natural product research community. So that kind of demonstrates the broad range of compounds that you can approach. A lot of these natural products that we've looked at are much more flexible than the systems shown. It's just the ones that are shown are published. If we want to take this further, our ideal situation is to push this technology a little bit harder, and one of those options was to try and go directly from HPLC into crystallization. And this was a project in collaboration with Jem Stach, where analysis of a compound gave fragments that had the wrong mass for anything that was expected to be in the system. And so from a small amount of material, we analyzed the material and resolved the structure. Had nothing to do with what they were expecting, but gave full resolution and was an unknown structure as well. So it shows that the potential for this is there to go almost direct from HPLC to crystallization work to full crystal structure analysis. If we then look at a summary, I guess we could kind of go through these things and say that this encapsulated nanodroplet technology seems to work very, very well. Its solution phase gives access to things which classically have not been accessible through solution phase in some cases. It's a semi-automated setup. We've got much higher throughput, so we can do parallel screening. The amount of material we require is really quite small. We have a wide range of variables that we can approach to alter the success rates of our crystallization, and all the materials we get out which we can grow crystals for, we we can analyze by single crystal X-ray diffraction. We've definitely got access to polymorph screening. We have other clear examples of this other than Roy. I just haven't had time to talk about today. And we've approached these challenging or uncrystallizable molecules, with the example being the dithionon shown. And we can definitely work with sample limited materials like the natural products. So I guess from a sales pitch point of view, we could go that Enact is an ideal technology for crystallization exploration. We can approach academic collaborations very easily from using the National Crystallography Service within the UK, or direct contact with us if you're outside the UK. The technology is patent protected, so for industrial access you can either come via the National Crystallography Service, again through me, or through our spin-out company Indicatrix Crystallography. And that almost brings me to the end, but I need to just highlight the situation where we have a full understanding, or not quite, of exactly what's going on. So if we watch this little video playing where we have our droplet having moved slightly within the oil to one side, and we're awaiting crystallization here and still awaiting crystallization until we notice that the crystal is actually growing at the oil air boundary here. So we've had diffusion of our sample through the oil, hit a phase boundary with the oil and the air, has initiated crystallization, and we get crystal growth. And we get crystal growth on this top edge as well, starting. So quite a way away from where we were expecting our crystallization to occur. But at this point we don't really worry about that, because we still successfully got a crystallization of the compound we're looking for. And in the end, that is the name of the game. So then really we should look at how this has impacted things from a global sense. We've had success with industrial collaboration, so looking at a new polymorph of CBD. This paper was out just earlier this year. We've got academic work going on in several senses, in the natural product and in the straight organic chemistry arena. We've even managed to get involved with some COVID work, and I said quite a bit at the beginning about different crystallization techniques. So if people want an overview of the crystallization approaches that are available, I can recommend this tutorial review by Jess Metherell. It's incredibly good. And that just leaves me to thank all the people that were involved and funders, and particularly you for listening. I will take any questions that you have, either now or in the future.
Okay, thank you, Mike. That was a very clear and precise talk. I'm sure you'll all agree. We'll move on to the questions. There are some. Don't forget to ask them in the chat if you haven't already done so. First question of the day is, based on the Y19 structure of Roy, Mike, you mentioned seeding to achieve that one. How does that work with Enact?
So it's a great question. There are different ways we can seed the crystallization experiment. We can either take our seeds, so assuming we've got a seed stock to start with, and dispense them in the oil itself, so that gets dispensed across the plate just as in part of the oil. We could streak seed across the plate, so just literally taking a needle and streaking the seeds across the plate through those droplets to give localized concentrations of them. The options here are quite varied. We can get the seeds into the crystallization condition relatively easily, and it does definitely increase the success rate of the growth of higher quality crystals.
Okay, thank you. Moving on. Is the system able to work with air-sensitive compounds?
So depending on the sensitivity of the compound, yes. It's easy enough to work with dry solvents. One of the options that we're currently exploring is to house a crystallization system within a glove box, so it's entirely anaerobic in terms of the conditions, with dry solvents throughout. And at that point, there should be no issue at all. The advantage here is setting up stock solutions doesn't require so much dexterity with glove work as setting up manual crystallizations, and because the robot does all of the tricky work, as long as the robot's inside the glove box, everything should be fine. So that is one direction we are heading in, but we currently don't have the hardware for that, but we can operate with things that are moderately sensitive already.
Okay, cool. Okay, a bit more of an ethereal one for you. Given the data generated, do you think it will ever be possible to predict the conditions for crystallization of given the molecular information?
So
good luck with that one.
Yeah, it's an interesting idea. So classically, I think most crystallographers have decided that you can't predict the crystallization conditions for a given compound. We're kind of sitting on the fence at the moment. We can definitely say already that particular compounds won't crystallize from particular conditions. So we have statistical evidence to prove this. So we can definitely say which conditions you should avoid if you've got a particular compound class. At the moment, we can't predict exactly what the conditions should be, but what we do think we will be able to do with the amount of metadata we get from the crystallization conditions, because we're not just trying one condition until it fails, we're trying many conditions, so we get a large amount of failure data as well as success. We think we will be able to narrow down the region of crystallization space in the relatively near future so that the success rate becomes higher, and also the amount of material becomes lower because you don't have to explore regions of crystallization space that won't work.
Okay, good answer. Okay, another one. From a cold start, how many experiments do you roughly need to do to, it's a bit of an open-ended one again, to get a result?
So
how long is a piece of string bike?
Yeah, so my facetious answer is twice as many as half the amount we need. In reality, our initial screening process uses 12 solvents dispersed across sort of chemical space. So they have different pKa points and different volatilities. So we have these 12 standard solvents that we use that are listed in the paper if anybody wants to go and look them up. And those give us quite a big insight into where we should go next. So we're looking at around about 400 crystallization experiments with very minimal sample usage to explore those 12 solvents. And then we can expand out depending on how many we need to go to. So our sort of more full screen at the moment is around about 90 odd solvents, but that's before you even start thinking about the conditions with anti-solvents and secondary conditions put in as well. So it is definitely an open question with an open answer. We usually have an idea about how long it's going to take from the first screen. If we start seeing success, we know we're going to get there, and then we tend to use derivatives of the solvents that have given some success.
Okay, very good. Last one, I think we'll call it a day after this one. Looks like a pharma-based question. Can the technology be licensed to use in-house?
It can. So the technology at the moment is exclusive, or the patent is held by the company or the university, and then an independent license to the company. So if people would like to explore that, then they should get in touch with either myself or Dr. Mike Hall at Newcastle, or via the company, and we can discuss that. It's definitely something we are open to. Yeah, just get in touch.
Okay, jolly good. Well, obviously, we'll have to thank you. I'd like to thank Mike again for his very good talk. Obviously, we'll provide a link to the webinar for those people who've perhaps missed it for whatever reason, or if you want to review it in the comfort of your own home. Later on, we'll send that out in a couple of days. So yes, thank you, Mike, again. Thank you to you, the people who've attended, for making time in your busy days for this. I hope you found it useful, and we all look forward to seeing you soon. So thanks again, and take care and enjoy the rest of the day.
Thanks
Bye.
very much.